Chapter 4 is tissues. And just by looking at this image here, we can see that there are a number of different kinds of tissues that make up the body. And these are the four big groups of tissues: nervous tissue, muscle tissue, epithelial, and connective. And each one of those categories has multiple examples of different types of tissues within it. So the focus of this chapter will be looking at those different tissues. The first type of tissue we want to look at is epithelial tissue. And I really like epithelial tissue because this is what makes up your skin. And as you'll find in Chapter 5, I have a great interest in skin. So we can classify the different kinds of epithelia based on a couple of different things. And the first one is its cell type in terms of thickness. Is it a simple tissue, or is it a stratified tissue? So a simple tissue is one that has a single layer of cells. A stratified tissue, as you can see in the image, would have multiple layers of cells. We can also classify epithelia based on shape. And epithelia come in three basic shapes. The first one is squamous, then cuboidal, and columnar. And the squamous, really squamous means flat. Cuboidal means more of a rectangular shape. And columnar is, as this name suggests, a column type of shape, so it's longer than it is any other direction. So we could then say that you could have a simple squamous tissue, and what that would mean is a single layer of squamous cells. Or we could have simple cuboidal, or we could have stratified squamous. So that would mean in that case multiple layers of flat cells. So those are ways we can describe epithelia based on shape and based on number of cell layers that it happens to have. There's a little closer up look at the simple versus the stratified there. So for example, our skin on the outer surface of the body would be a stratified type of epithelia. And if we were looking at lung tissue would be a simple epithelia. So where it is and what its job happens to be will depend or will determine what kind of thickness it needs to have, whether it needs to be a single cell thick or whether it needs to be multiple layers. Again, the different shapes there. When we talk about glands in the body, we can talk about endocrine glands and exocrine glands. And right now we're going to talk about exocrine glands because those are the ones that are found within epithelia of the body. And as you can see here, they come in a number of different flavors. They can be tubular in nature or alveolar in nature, and that's really referring to the shape. Is it a tube shape or is it an alveolar shape, meaning more of a bulb or ball shape at the end of the tubing? So within tubular, we can have simple tubular. So simple, again, typically means one. So the simple duct structure, the duct itself doesn't branch. So simple tubular, just one. Simple branched tubular is still a simple duct because the duct itself, that's the yellowish cells, did not branch, but the tubular structures at the end of it did branch. So simple branched tubular. Then we can have compound tubular, and it becomes even more complex. So the compound means that the duct itself, the yellow cells, did split into multiple ducts. And then within each of those, the tubules also probably split off from the ducts. So it gets bigger and more complex. And typically, the bigger and more complex it gets, the more of its product we need in a shorter amount of time. So if you're a very small exocrine gland, you can't make a lot of product and you can't do so quickly. But if you're a large, very complex, very branched duct system, then you can make a lot of product and you can probably do so fairly quickly. When we look at the alveolar structures across the bottom, the same concept applies. Simple alveolar means one duct, with the ending structure being more of the balloon or ball shape. Simple branched alveolar has multiple little balloons at the end of each duct. Compound alveolar would be just like the compound tubular, multiple ducts, multiple alveolar endings at the end of each one. And then to get really complicated, we can have a compound tubulo-alveolar, meaning the duct is branched in multiple branches. At the end of each duct, we have multiple endings, and we have both tubular endings and alveolar endings. So every concept we've seen so far here in exocrine glands, all in one. If we look at some of the different examples, we start to see their significance. So an intestinal gland wouldn't necessarily need to make a lot of product and wouldn't need to do so quickly. So simple tubular will do the trick. If we look at stomach glands, so this would make things like stomach acid, compared to, let's say, mammary glands or salivary glands, the mammary glands would produce milk, salivary glands produce saliva, and they would be needed to produce their product in large quantities on relatively short notice. Whereas your stomach has lots of time to make stomach acid, not a huge rush there, but when that baby wants to eat, the mammary glands had better have milk ready, and they'd better be able to make a bunch of it quickly. Also with the salivary glands, as soon as you get the idea of eating, even if you're not actually going to do so, those glands get activated. So you can't just sit around and hold the food in your mouth all day waiting for saliva to get there. So you need those very large branching structures so that you can get lots of product very quickly. There are two different ways that exocrine glands can release their product. So let's say here the pink cells are making the product, whatever type of product it happens to be. And I guess we should say here that the difference between exocrine glands and endocrine glands is what they do with their product. So an endocrine gland is going to release its product into bodily fluids. Exocrine glands dump their product onto the surface of the epithelium that they're associated with, or into a body cavity, but not necessarily out into bodily fluids, which means that their actions probably would be a little bit more localized than would be the endocrine gland dumping things into the blood, which would go everywhere. So our two types of exocrine glands here, our first example A is a merocrine gland, and it releases its product into the cavity of the gland by a process called exocytosis. Basically bundling up its product in little pouches and then releasing those by fusing with the outer membrane of the secretory cells. In a holocrine gland, the cell makes its product but doesn't release it by fusing with the membrane, but rather the entire cell dies, basically by exploding, and then releases its contents because it broke into pieces, and its internal contents are released into the cavity at that point. So a question I would have for you at this point is, which of these two glands would be more likely to develop cancer? A merocrine gland or a holocrine gland? So think about that for a minute. And it's a 50-50 shot, so take a guess. But also think about why you're choosing that particular one. Perhaps you have an answer by now. So if you voted for holocrine gland, you would be correct. The more important question is, though, why would a holocrine gland be more likely to become cancerous than a merocrine gland? And the answer is quite simple. It has to do with the rate of cell division, or mitosis. So the faster you're making new cells, just like the faster you do anything, the more likely you are to make a mistake. With holocrine glands releasing their product by exploding the cell, you obviously have to replace it. So an active holocrine gland must be constantly generating new cells to replace the ones that have exploded and released their contents. Merocrine glands can reuse the same cell over and over again, and the lowered rate of cell division then means it's less likely to make a mistake. It doesn't do it as often and it doesn't do it as quickly. So holocrine glands would be the more likely cancer source of these two. When we think about epithelium, some of its job is to be a lining for things. And there are several different kinds of, we would call them membranes at this point, lining epithelium. And our first example here is a cutaneous membrane. And a good example of a cutaneous membrane is your skin. And its job is to be a covering for a body surface, in this case the outer body surface. So it's typically a dry, very sturdy kind of membrane, and its job really is to keep things out of the body that don't need to come in the body. Example B is a mucous membrane, and mucous membranes also line surfaces, but they're going to line inner surfaces of the body, specifically openings to the body. So every external opening to the body has a mucous membrane associated with it here. And they show the openings for the respiratory and digestive system here, so both of those systems will be lined with mucous membranes. They're also hopefully going to provide some degree of protection against things that should not enter the body that came in through those openings, whether it's the mouth or nose. But they are thinner, so it is easier for pathogens to penetrate those surfaces. So most of the time when you pick up any particular disease from someone else or some environmental exposure, most of the time it was able to penetrate your system through mucous membranes. So let's say the HIV virus. You could have the surface of your body, the cutaneous membrane of the skin, covered in HIV virus, but if the skin was not compromised, then the virus would be unable to penetrate that skin. If, however, the virus enters the body and comes in contact with a mucous membrane, it would be quite capable of penetrating the mucous membrane and then entering the body. So these are weak spots, I guess you could say, in our protection from the external environment. The reason why they're weak spots is A, they're thinner, and B, they are always moist. So this is where the mucus part comes from. It's always going to be coated in a thin layer of mucus because those tissues need to remain moist. And if they become dry, then they start to lose their integrity and they start to perhaps die. So they're designed to be moist, so a wet surface that is a relatively thin surface is a prime place for pathogens to live and have opportunity to penetrate the body. Option C here are serous membranes, and their particular function is to lubricate. So you see here the parietal pericardium and the visceral pericardium. Basically, that's just saying that it's a lining around the, in this case, the heart. Then you would have a parietal pleura and a visceral pleura. Those are linings of the around the lungs and on the inside of the rib cage. You would also see them down in the abdominal cavity, basically surrounding every internal organ. And their job really is to provide a slippery surface so that organs don't stick to each other. You don't want the internal parts of the body sticking and then not being freely mobile. So the serous membranes help to lubricate and keep everything nice and slippery and movable inside. All tissue has the potential to be damaged. So let's talk about the process by which tissues repair themselves. In this particular example is the skin, but really all tissue will work pretty much the same way here. Let's say you cut your finger. It's going to bleed, and it's going to bleed because blood vessels in the skin were damaged and then blood came out. So bleeding is typically the first step in tissue repair. And at this point, bleeding does two things, and you would typically want an injury to tissue to bleed for two reasons. One is that it cleans out the wound. So whatever it was you cut yourself on here may have introduced some bacteria deep into that cut. There may also have been bacteria on your skin that were not able to get into the body, but now this cut is a wide open entrance, and they might try to take advantage of that. So if that injury bleeds, the bleeding will probably wash out all of the pathogens. A number of years ago, I was mowing the lawn and stepped on a nail, and it went almost all the way through one of my toes, and it would seem like a potentially very infectious problem because the nail's laying there in the nasty dirt. Now it's been penetrated up into my toe, and not just like a cut or scrape, but it would be considered a dirty wound. And a dirty wound is one that is not easily cleaned out by bleeding, because in this case a puncture means that the external opening is small and the blood would have a hard time coming out through that smaller opening. It happened to bleed a lot though, and it cleaned out very nicely, and I had no infection whatsoever from that. So that was a good example of bleeding and the benefit that it provides. You do, however, not want to bleed forever, so you do need to have that bleeding stop. And when a scab forms and a blood clot forms, then the bleeding will stop. And that typically occurs within a couple of minutes for a standard cut or scrape. I think that puncture on my toe probably bled for about 20 minutes, but that was because it was a relatively good-sized blood vessel that it tapped into when it poked in, and it took a little bit longer for that one to seal over. After you stop the bleeding, then an inflammatory response occurs. So inflammation is normal and good as a part of tissue repair. And a lot of times when we experience inflammation, we instinctively want to turn it off. We want to take an anti-inflammatory. And I would suggest that we only do that in cases where it is interfering with reasonable body function. But the point of inflammation really is to isolate that area of injury. So if there is a pathogen that didn't get washed out with the blood, the swelling that occurs in that area would wall off the area from the rest of the body. If an infection does occur, it is then a local infection, and hopefully with appropriate inflammation would not become a systemic infection. Turning off the inflammation would allow potential pathogens to spread more easily through the body. You can also see here some white blood cells getting out of the blood vessels, so they're actually squeezing out of these very small arteries here into that area to help with the cleanup process. So really, step one of tissue repair is all about cleaning. Bleed it out, inflame it so anything left can't get easily out of that area, and then dump white blood cells in to clean up whatever everything else messed. Step two is restoration of blood supply. So any vessels that were damaged in this process will regrow and repair. And you're also going to see, in this case, the outer surface of the skin would start to repair itself, and an accumulation of what's labeled here as a fibroblast. Basically, this is the formation of scar tissue. So scar tissue comes in and ties all this damaged tissue together, holding it in place, and allows the tissue that was there in the first place to rebuild itself. So it's sort of like internal biological stitches to hold things together and give them an opportunity to heal. And every time you have tissue injury, you have scar formation. Most tissue injuries don't result in visible scar tissue. Most of the time it's a small enough amount that it holds it together but doesn't have any particular visibility along with it. Some people's bodies, however, produce excessive amounts of scar tissue, and my body happens to be one of those. So if I have something like scraping my knuckles, normally for an average person you would bleed, the skin would regrow, and life would go on. For me, I would have visible masses of scar tissue left behind because my body produces so much scar tissue trying to tie things back together that it can't shrink to an invisible level. So that scar tissue will always remain there even after the injury is long healed. But in general, it was a small enough amount, and as it ages, it shrinks a little bit, and for the most part, you don't see it. For me, it's large amounts, and because it was such a large amount, even though it does shrink over time, the surrounding skin is not capable of covering it over and making it appear that nothing ever happened. So if a standard scratch or scrape does that to me, you can imagine what would happen if a doctor would surgically slice into me, making a very large wound track. And I've had to have that many times with potential skin cancers being removed. So every time that happens, it results in large visible masses of scar tissue. Even though the last time I had it done, it was done by a plastic surgeon, it still resulted in large amounts of visible scar tissue that weren't really any better than any of the other times I'd had it done by a non-plastic surgeon. So my skin, my tissues overreact in terms of producing scar tissue. Sometimes you hear people talk about keloid scars. That's exactly what this is. That just means you produce too much and it remains visible forever. Step 3, the scar tissue starts to shrink. The blood vessels that were damaged are repaired, and the coating around that particular tissue is probably pretty much regenerated. So if you think about cutting your finger, you might say that would take 1 to 2 weeks for complete regeneration and repair to occur, such that you would, again, typically not notice that anything had ever happened. What I have noticed as I age is that my tissues repair more slowly than they once did. And that's probably true for everybody. The older we get, the less enthusiastically our body puts itself back together. So what normally would take about a week to heal itself, to disappear like nothing ever happened, now takes a little bit closer to 2 weeks. So getting older certainly has very little in the way of positive associated with it, but unfortunately it is also unavoidable. So that's the process of tissue repair. All the different tissues in the body come from 3 basic layers of tissue. So what you see here is a mass of cells that they have very kindly colored as blue, red, and yellow cells. And this is what a 16-day-old human baby would look like. So 16 days after the egg was fertilized by the sperm. And at this point, it doesn't at all look like a human, but the 3 different, what we would call germ layers, have been formed. And each of these layers then will form other more specialized tissues. So the first time we see distinct tissue being formed in a developing embryo is at this stage. So the ectoderm, mesoderm, and endoderm then specialize later into other specific things. And here they say that the blue will give nervous tissue, the yellow will give digestive system, and the red will give muscle and connective tissue. Each of those 3 things also become other things other than what's listed here, but the point is everything, and then there are dozens of different kinds of tissue in the body. All of those different kinds of tissue really originate from one tissue type, which became, before 16 days, became these 3 types, and then differentiated into the very diverse types that make up the body. So even though these tissue types that you're going to see throughout Chapter 4 are very diverse in appearance and in function, they really all came from the same basic cells that just specialized later on in development. As you will notice as you look through Chapter 4, there are lots and lots of different tissues discussed here. So what I would like you to do is not necessarily spend a lot of time on this, but go through and look at all the different tissue types. So there's a picture for each, there are drawings for each, and a little bit of a discussion on what they're like, what they do, and where you find it. So just flip through the chapter and look at all those for a few minutes. And what I would like you to do is develop an appreciation for the vast complexity of tissue types that make you up and that do what they do. And I think that if you consider all those tissue types for a little while, you'll be quite impressed with the diversity of cell and tissue types that we have, the diversity of function that we're able to have, and remember, it all came from one original tissue. Hopefully you found Chapter 4 informative and useful, and I'll see you next time.